Alright, let's see what happens when we do the experiment. Here I have a syringe filled with purple grape soda. I'm going to push the grape soda into the column and let the liquid go through into cup number 1. Now as I do that, the water is going to go through the column, but the dyes that are dissolved in the water are going to become trapped in the column. The column is designed to be sticky to the dyes, but not to be sticky to water. So water goes right through. And the liquid that's coming through is mostly clear because the dyes are getting stuck to the material inside the column. So then what I'll do is I'll try to rinse out the materials from the column using the two different mixtures of water and alcohol. First with the 5% alcohol and then with the 25% alcohol. Okay, so the water that went through, mostly clear, and now the dyes are trapped inside the column. Now I'll take the column off and fill the syringe with 10 milliliters of 5% isopropanol, so it's 95% water. And as I squeeze this through, it's going to wash out one part of the mixture, but not the other. The color that's coming out first, you can see there's a red color there. That is red dye number 40. If you look at the ingredient labels on foods that have food colorings in them, they tell you what the name is of the dye. Now there's another color that's in the soda. To give it the purple color, there's a mixture of red and blue. The blue dye is not going through the column right now. It's staying stuck because it's not attracted as much to the liquid that's going through the column. The liquid going through the column dissolves the red molecule, but not the blue molecule. So now I'm going to use the other mixture of water and alcohol. This one has more alcohol in it. It has 25% alcohol. So it's going to dissolve differently the materials that are inside the column, trapped in the column. So there's a little bit of red still left, but now it's coming out blue. This is blue dye number 1. Again, that's from the label for the soda. So the two different dyes are soluble in water, but they're also different in their solubility in the alcohol. The first dye is more soluble in water. The second dye is a little bit more soluble in alcohol. So when you increase the amount of alcohol in the mixture going through the column, it dissolves better and washes out. So the first cup, pretty clear. The second cup, red dye number 40. And the third cup, blue dye number 1. Okay, let's try that with the green Gatorade. So I'm going to push the green Gatorade into the column, and again the liquid will go through and the dye will be trapped inside the column. The liquid coming through is mostly clear. A little bit of one of the dyes might sneak through, but for the most part the dye is staying trapped inside the column. Yeah, I see a little bit of yellow is going through first. That's unexpected. I really thought that the yellow dye would stay trapped inside the column more. But now I'll add the 5% isopropanol. There are two dyes in this mixture. The yellow dye is more soluble in water, and the 5% solution has more water, 95% water, 5% alcohol. So the liquid that's coming through is mostly yellow. The liquid that went into the first cup, since the drink contains mostly water, it's not a surprise that the dye went right through along with the water because it's very soluble in water. But when you look at the column, you can see there's a blue color that remains. That blue color is not as soluble in water. It's the same blue dye that you find in the purple drink. This yellow is yellow dye number 5, if you look at the label on the ingredient list. If I add the 25% isopropanol now, just like with the purple drink, this time out comes the blue dye. They're the same blue dye number 1. This time it doesn't look quite as blue because there's not as much dye. But if you had more dye in there, it would look darker blue. But on the list of ingredients, they are the same dye. So the dyes differ in how soluble they are in water compared to how soluble they are in alcohol. The more soluble the dye is in water, the earlier it comes out. The first time the pink or the red dye didn't come out because the water went right through and the dye was trapped in the column. But it did get washed out when 5% isopropanol was used. Less alcohol, more water. In this experiment, the water dissolves the yellow dye a little bit better than it dissolves the red dye. So that one goes right through along with the water. 5% alcohol takes the yellow dye out of the column. It's more soluble in water than in alcohol. And then in both of these, the blue dye comes out when there's more alcohol, because that dye is less soluble in water and more soluble in the alcohol. Now what we can do is we can compare the polarities of these two molecules and use that to tell which is a more polar or nonpolar dye. The red dye number 40, the blue dye number 1, the yellow dye number 5. Okay, let's take a look at the questions together. On the second page, I'll go through question 1. I'm going to skip question 2 for a moment, and then I'll look at questions 3, 4, and 5. I'm going to ask you to work on question 2 yourself, because next week we're going to test your answer to question number 2. So I really want you to think that one through before we try testing the answer to that one together. So let's look at the first question. It says, explain why the steps in the experiment were able to separate the dye mixtures into the different parts. What has to be different about the dyes to separate them, and why are different mixtures of water and isopropanol pushed through the column? So the two different parts of each of those mixtures, water and isopropanol, were what we used to try to wash out the different colored dyes from the column. Think back to our do now from yesterday. We were looking at examples of separating mixtures. One way that you could separate a mixture is by the difference in the particle size of the materials that are in the mixture. I might use this one to separate a solid from a liquid. If I'm trying to strain the noodles out of the pot of water, I might use this one to strain out the coffee grounds as water is going through. It might dissolve part of what's in the coffee, and then whatever can dissolve in the water would go through, leaving behind the solid material. So differences in particle size. Small particles can go through, large particles can't. Something has to be different about the two things that are in the mixture that you're trying to separate. We looked at another example yesterday in our do now of a stain in your pants. If we had a mud and grass stain in your pants and you try to wash it with water, if the water is able to wash away the mud but not the grass, then that means that there's something about the grass and the mud stain that's different with respect to their solubility, how they dissolve or mix or not with water. Water can mix with the mud part of the stain, but not the grass part of the stain, because they have attraction for each other, or they're similar in their attractive forces. So as the water goes through, it dissolves the mud, but not the grass. You might have to use something else to dissolve the grass stain. This is what's used at dry cleaners, a chemical called perchloroethylene. It dissolves the grass stain because it wasn't able to dissolve in the water. So to separate the two parts of the mixture of mud and grass requires two different solvents that have different attractions for the different parts of the mixture. Keep that in mind when you're thinking about answering that first question. What has to be different about the dyes, the red, the yellow, and the blue dyes, has to do with their solubility in the mixtures that were pushed through the column. The mixtures differ in how much of the water and how much of the alcohol they had. The first one has only 5% of the alcohol, and the second one has 25%. So it differs in how much water and alcohol are in the mixture. Skipping question 2 and looking at question 3. That molecules have a property called polarity, which describes their charge-sidedness. If we were to mix together some molecules, we've used some of these before. If I put water and the isopropanol together, we know those two mix because we used them yesterday. If I mix them with decane, though, one of the molecules we've used in our Why Do Things Mix experiment, those are able to mix decane and the alcohol, and we know from our previous experiment that decane and water don't mix. And so if I were to do the Venn diagram showing which ones can mix and attract to each other, it would look kind of like this. We did something similar in our previous experiment. So the question is asking about what does this mean about the polarities of the molecule? If we're given that water is very polar, if decane doesn't mix with water, then that tells us something about its polarity. We use a rule of thumb that likes dissolve likes. Things that are like in their polarity properties can attract to each other and stay mixed. If decane can't mix with water, that means that it's less polar or nonpolar. Polar and nonpolar are like tall and short. They're terms of comparison. So isopropanol, it can mix with water, so it must have some degree of polarity similar to water. It also mixes with decane, so it must have some degree of non-polarity like decane. So it is less polar than water. It's more polar than decane. So in terms of comparison, less polar than water, more polar than decane. Something about the mixture of water and isopropanol changes the polarity of that mixture because it has something in it that's less polar than the other part of the mixture, the water. So some features of these molecules that might play a role in the polarities, when you look at water and isopropanol, you'll see that they share in common the presence of oxygen bonded to hydrogen. And we know what that can do. That can make hydrogen bond attractions to other molecules that have similar oxygen bonded to hydrogen in their structure. The decane doesn't have any atoms of oxygen, so there's no place where water or isopropanol would be able to make hydrogen bond attractions. However, isopropanol and decane both have long chains of carbons bonded to hydrogens. And when you look at the symmetry of this, you'll see that they're very, very symmetrical. If I just look at this part of the isopropanol molecule, I see that it's mirror images on the sides, left and right, and it's mirror images top to bottom. Very, very symmetrical. And the same goes with the decane as well. So one feature that might explain why isopropanol and decane can mix is their symmetry and mostly carbon and hydrogen bonds. Something that might explain why water and isopropanol can mix is the presence of oxygen and the ability to make hydrogen bonds. The more isopropanol there is in the mixture, the more you have present a molecule that's less polar and not able to make hydrogen bonds as much as water can. It can make hydrogen bonds on two sides. The more water there is in the mixture, the more it's like water, more polar. The more alcohol there is in the mixture, the more it's like the alcohol, which is less polar. So I'm just going to put here that it's less polar. The fourth question is asking about the mixtures of water and isopropanol that were pushed through the column. They contained less or more isopropanol. How does that affect the polarity of the mixtures? That's part of our discussion here. More water in the mixture means more polarity because there's something present that is with more hydrogen bonding potential. Something that is less polar in the mixture, as you increase the amount of that, it becomes a less polar mixture. So the 5% solution is more like water. The 25% solution is more like isopropanol. The 5% solution is more polar. The 25% solution is less polar. So that tells us something about the polarities of the molecules that we're able to mix. So when there was more water in the mixture, the 5% solution, we saw that there were two of the molecules that were able to dissolve in that solution. The one that's 5% isopropanol, which is a greater concentration of water. The one that had more isopropanol, the 25% solution, has more of the alcohol, the less polar mixture. This one is more polar. This one is less polar. And the material that was able to mix with this was either the red or the yellow dye. So those are more like water. We could score them as being more polar because they're more like water, using the rule of thumb, likes dissolve likes. The blue dye is less polar. It doesn't mix with the solution that contains more water. It mixes with the solution that contains more isopropanol, the one that has a less polar molecule in greater percentage. The last question asks about how the polarities of the three dyes compare to each other. I think that's the question that we're answering here. And so as you're thinking this through, you want to look back at question number 2. Question number 2 is asking about what makes Kool-Aid orange. Is it one dye, an orange dye, or is it two dyes? Could it be a mixture of the red and yellow dyes that we saw in our experiment? If you were to use the same cups, the same solutions that we used in our experiment, would you be able to tell the difference between a one orange dye or a two yellow and red dye mixture? Why or why not? We'll test your answer when we get back to class together on Monday. Please make sure that your good work stays handy in your folder until we get back to class next week.